Hello, Bonnie Cohen. See you.
Hello,
All right.
I want to say hello to everybody that I haven't seen for so long. Hi, everybody.
All right, all. So let's get started. I'm sure people will still come in. I've got a few things, sort of housekeeping things to talk about and let people know what's coming up in the next, in the next month or so. So let me sort of start by welcoming everybody to our second CBA talks of the year. And before we talk about what we're doing today, let me give you a heads up on some of the amazing programming that we've got coming up. Claire, we were just talking about how neat it is to kind of be here and be doing things, and there's lots of opportunities. So our next and final CBA talks for this year is going to take place on April 18th, so about a month from now, a little over a month from now. It really is going to be an interesting talk with Dr. John Joel Cully, one of our longtime members. And that's, by the way, for those people who haven't figured out yet, CBA Talks is all about our members. And yes, we can bring in and we can hire all sorts of speakers from outside, but we have some really amazing people within our congregation. And for these last couple of years, we've been working on trying to expose our congregants to the people within our congregation. So we've got that coming up in April, as well as today. It's called Strangers in the Land. And Jay is going to talk about sort of the other side, the not so well-known side of the internment camps that happened during World War II. Most of us know about the Japanese internment camps, but there was actually internment that was happening beforehand. And I had no idea about this until Jay actually shared it with me. And he's done a lot of research on this. It's going to be very, very exciting. So I encourage you to register for that and sign up. That'll be, as I said, on April 18th, also at 10 o'clock. A couple of other things coming up. Our CBA Cafe, our movie nights, has been something that during COVID, we really struggled to figure out how to do. We can't get all together. We can't figure out how to bring groups together. So what we're going to do instead is we're going to let people watch a movie, or in this case, a series, and then come together as a community to talk about it. We're going to be looking at the series Unorthodox. You can find it now on Netflix. For those who haven't seen it, please, what we're going to do is ask you to watch the first 2 episodes. We're going to talk about the first 2 episodes on March 16th, and then watch the second 2 episodes, and we're going to talk about those on March 23rd. So it gives people an opportunity to see something, and then we'll all get together to be able to discuss and talk about them. That's going to be our CBA Cafe. For those who have not yet experienced our Zimratia Malavamakas that we do about once a month on Saturday nights, highly encourage you to join us. It's a great way to end Shabbat. We start with Havdalah and then sing together. The next one is going to be March 20th. It's a little bit later than what we've done before. They'll start at 8 o'clock p.m. because now we're going to be changing our times and Shabbat is going to end a little bit later. You can see it on our live stream, and you can see it on Facebook Live. And finally, we were just finishing our Book of Job class. Many of you are in the Book of Job class that we've been doing on Tuesday evenings. In April, we're going to start our 3rd class of the year. I always try in the 3rd class to do something that has to do with spirituality. And so starting April 6th, Tuesdays at 7:30, we are going to be doing the spirituality of Heschel, Abraham Joshua Heschel, and looking at some of his readings and really trying to figure out, the spirituality that he brought to us in the 20th century. It's amazing. So I encourage you to join us for that. For most of these things, you do need to register in order to get the Zoom link. So please register on our website on the calendar. Once you do that, you'll get the Zoom link with the passwords and things like that. Okay, that's what the upcoming, but now let's talk about today. We have an incredible opportunity to hear from one of our congregants today, Dr. Darren Siegel. Dr. Siegel treats patients with a variety of GI malignancies, including esophageal and gastric cancer, hepatobiliary cancer, pancreatic cancer, colorectal cancer, and neuroendocrine tumors. He has a particular interest in pancreatic cancer and has helped develop a multidisciplinary pancreatic and biliary cancer program at Scripps Clinic. He has created a transitional GI cancer research program with collaborations with the local and regional biotechnology industry. His areas of research focus on predictive biomarkers, novel therapeutics, and immune therapy. And Dr. Siegel actively accrues patients to both novel investigator-initiated and pharmaceutical-sponsored trials, which means he's on the cutting edge of what is going on in cancer research today. He also reviews and publishes his research in major clinical and scientific journals. He graduated with honors, very high honors from UCLA with a degree in microbiology and molecular genetics. He's earned his medical degree from the UCLA School of Medicine and completed his residency in internal medicine at UC Davis School of Medicine. Upon finishing his fellowship in the Hematology Oncology program at Scripps Clinic, he joined the Division of Hematology Oncology at Scripps Clinic and is now the director of the GI cancer program at Scripps MD Anderson Cancer Center and Scripps Clinic. But more importantly than all of that, he and his family have been Beth Am members and very active Beth Am members for many, many years, enriching our community in so many different ways. And it is my pleasure to introduce Dr. Darren Siegel to speak today about the beginning to win the war on cancer. He titled it Sending in the Troops. And Dr. Darren Siegel, if you would please, I'm going to spotlight you.
Okay.
And it's all over to you. Welcome.
Okay. Thank you, Rabbi. I'm going to share my screen. Give me one second. Here we go. Okay, can everybody see? Okay, perfect. So I wanted to thank Rabbi Kornberg and Susan for asking me to put this talk together and deliver it to our community. I think it's so important and special to be able to do this during the time of COVID, especially, and you know, and maintain the closeness and the relationships that we all have built at Beth Am. So thank you for the very nice bio, Rabbi Kornberg. The title of the talk today, as was already mentioned, is Send in the T cells, the troops, beginning to win the war on cancer. Hold on. Am I able to advance the slides, Rabbi?
If you go down to the bottom there, to the arrow.
Oh, here we go. There we go. Got it.
There you go.
Thank you. I have a variety of slides. I think we can get through them in about 45 minutes. I want to leave time for questions. I'm happy to go beyond an hour if there's questions. And you know, and you guys can send notes to the rabbi as I'm talking. You know, no reason to. Feel free to interrupt me. The talk is going to flow first with an introduction to cancer medicine. We're going to look at melanoma. My main expertise is pancreatic cancer and pancreatic, biliary, colorectal cancers, but really melanoma is the paradigm for immunotherapy, as well as the futility of standard cytotoxic chemotherapy historically. To understand what we're talking about, we all need to become immunologists today, and I'll have a brief primer on immunology. Then we're going to dive much more deeply into the focus of the talk of immunotherapy, specifically immune checkpoint inhibitors. There's a variety of immunotherapies that exist today, but these are the dominant ones and really the ones that have been game-changing as it relates to solid tumors. So again, we're going to focus on melanoma, but then we're going to touch on the role of immune checkpoint inhibitors in other solid tumors, the ones that I know much better. And then, you know, pancreatic cancer, unfortunately, some of the tumor types that I take care of haven't benefited from the really, truly profound advances as other tumor types have. And we're going to talk about some areas of need for further research. I have a variety of de-identified CT scans that I thought would be really beautiful examples to look at. and show the power of immunotherapy, and then, formal questions. So, up until the past 10 to 15 years, treating cancer was very similar. I would imagine a medical oncologist would have felt very similar to the way Don Quixote felt trying to defeat the windmills. When I finished fellowship in 2008, you could kind of measure and really understand any tumor type, breast cancer, pancreatic cancer, lung cancer, whatever the case may be. You could really understand the cutting edge area of research by maybe reading 5 to 10 papers. Anybody on this call could read 5 to 10 manuscripts and really know as much as their treating physician would know. But over the past 10 years, there's been an incredible proliferation of research and true clinical advances that have made it much more complex. But really, up until the past 10 to 15 years, the benefit of treating cancer was debatable. The first cytotoxic chemotherapies were developed in the early 1940s. We could talk about the stories of World War II, where troops were exposed to nitrogen mustard agents that then were who lost their bone marrow function, and it was understood that these molecules could have therapeutic benefit, and they were deployed in pediatric patients with leukemia, all tumor types, and there was initial incredible excitement in the 50s and early 60s that these tumors were going into remission, but was quickly understood was that these remissions were very short-lived, and very few people were cured of cancer. Metastatic solid tumors, even today, generally are not felt to be curable, but even 40, 50 years ago, there were only very rare examples, germ cell tumors, testicular cancer, choriocarcinomas that could be actually cured. The story of pediatric leukemia was much more promising, and that is clearly one example of the benefit of chemotherapy even early on. But for the most part, the story of treating cancer was one of nihilism and despair. So I wanted to highlight this with some, I think, interesting and somewhat funny statements that I found through my reading. Dr. Paul Ehrlich was a very famous German Jewish scientist, immunologist. He won the Nobel Prize in 1908. We'll talk more about him as the talk proceeds. He theorized antibodies. He created the first therapy for syphilis. So he was a giant, and he was a scientist. And above his laboratory door, as you entered, he had a sign that read, "Give up all hope, O ye who enter." So, I think a guy who really was moving the field forward 100 years ago had this sign above his door. There's another a review article I read from a man named DeVita. The reference is below. And give me 1 second, just want to shorten my picture so I can actually see the whole screen. So DeVita was also a giant. He developed the first cure for Hodgkin's disease, coming up with something called MOP, M-O-P-P, that was replaced by something called ABVD. In any event, he also is a historian of medical oncology. He authored the key textbook when there were textbooks. Now everything's online with UpToDate, but he had the DeVita text. And this specific sentence really spoke to me. In the 1960s, medical oncology did not exist as a clinical specialty. Those who were given the task of administering chemotherapy at most medical centers were regarded as underachievers at best. The main issue of the day was whether cancer drugs caused more harm than good, and talk of curing cancer with drugs was not considered compatible with sanity. So first, I think my mom's on the line, and I'm sorry that she realizes I'm now an underachiever, and it may also, I don't want it to taint the rest of this discussion, but it really speaks to the fact that treating cancer was difficult. You had to be a true believer to pursue that because success was minimal. Sorry. These are the 5 what are called pillars of cancer therapy. Now, again, up until the past 15 or 20 years, all we had were the first three: surgery, radiotherapy, and cytotoxic chemotherapy. And that's really why success was limited to people that did not have metastatic disease, was limited to people that could tolerate chemotherapy, and very few lucky patients with metastatic cancer who were exceptional responders. Now, surgery existed at the time of Galen and Rufus 2,000 years ago, maybe even with ancient Egyptians. I don't think anybody would have even wanted surgery over 100 years ago. But today, surgery still is the primary modality to cure patients that have localized cancer that has not spread. Surgery is still the central intervention to do that, but not it's changing. Radiotherapy is radiation treatment, usually external beam outside the body. Proton therapy, you hear about IMRT, SBRT. There's a lot of different techniques, but radiotherapy is still like surgery. It's a localized technique. If you have metastatic cancer, for the most part, radiotherapy will not have a role because you can't radiate the whole body. Cytotoxic chemotherapy, that's what we've already been speaking about, came out of generally World War II and some of the research centers at Yale. And there's dozens, maybe hundreds of cytotoxic chemotherapeutic agents that can improve cure after surgery. It's called adjuvant chemotherapy, and can produce responses and prolong survival when somebody has metastatic disease, but you're generally not going to be cured with cytotoxic chemotherapy. Things really started changing in the mid to late 90s with the advent of Gleevec or imatinib. Targeted small molecule therapies are called tyrosine kinase inhibitors that are designed to specifically interfere with a driver genetic mutation in the tumor. So understanding the biology of cancer became the understanding of the biology of cancer became much more prevalent with more powerful genetic tools to sequence the tumor DNA with next generation sequencing, and the variety of advances since then. The point is, there are certain genetic changes that drive certain tumors. The classic one is c-KIT, another one, the Philadelphia chromosome. There are oral tablets that specifically target those mutations and really have a profound impact on that disease, but it's still generally not a cure. People need to stay on these, and sometimes you develop resistance. And so again, it's another big advance, but it doesn't deal with the global problem of cancer, which is not just the genetics, it's the stroma, it's the body's response to cancer, it's the soil, the connective tissue, the cells that the body that react to the tumor, that sometimes drive the tumor and protect the tumor. Now, immunotherapy came around in the really late 90s, but really it was the mid 2000s that it found its true place, because the treatments became much less toxic and much more powerful and effective. And immunotherapy, in many ways, is the holy grail to treating cancer. So immunotherapy has a variety of ways to do it. Cell therapies, we may have heard of CAR T cells. They're very effective in leukemias and some lymphomas. There are better or more emerging, newer cells such as NK cells, CAR NK cells that are coming as well. Cytokines, something called interleukin-2. Interleukin-2 we'll talk more about in a few more slides, but that was the first approved immunotherapy in melanoma. It is a cytokine. It's a cell. It's a molecule that promotes the proliferation of cytotoxic cells and helper T cells, but it's very toxic. There's something called dendritic cells that can be manipulated. There's a drug approved for prostate cancer that was one of the early approved immunotherapies as well. There's cancer vaccines that in solid tumors have not had a huge impact, but today we're going to speak about immune checkpoint inhibitor therapies, which are essentially monoclonal antibodies. Before we get into that, I want to introduce the concept. I want to introduce the state of melanoma therapy in the year 2000, so 20 years ago, and the fact that all that we had for melanoma, and almost all other tumors, was chemotherapy. And this, very depressing result. This study was considered somewhat of a minor victory. And it just goes to show you how limited the therapeutic options were and the low expectations physicians and patients had. So this was a randomized phase 3 study looking at a molecule called temozolomide versus another molecule called dacarbazine in patients who had untreated metastatic melanoma. So just as a brief overview, whenever you look at a study, you need to understand what's the phase. Is it phase 1, phase 2, phase 3? Phase 3 is the most advanced. It's always randomized to look at the benefit, to prove the statistical benefit of a new drug versus the prior standard of care. You want to know how many patients were involved. Was it a small study? Was it a big study? Obviously, big studies are more valid and applicable. Was it randomized? How was the randomization? Was it 2 to 1, 1 to 1? What's the primary endpoint? The primary endpoint really determines how the study's designed, the statistics, how many patients need to be included. Do they pick an appropriate primary endpoint? And then, the outcomes and the p-value. Here, the p-value, you can see at the bottom of the screen, NS. Actually, I'm going to turn on my pointer. Here we go. One sec, here we go. All right. So NS means not statistically significant, so no difference. But it was a victory. Why was it a victory? Temozolomide is an oral chemotherapy tablet. Dacarbazine is the IV formulation. That's how people used to be treated with a melanoma. This was a randomized study of 305 patients. The primary endpoint was overall survival. Overall survival is always the best primary endpoint in medical oncology. If you look at the left-hand graph, this is what's called the Kaplan-Meier curve. A Kaplan-Meier curve estimates survival over time. And you can see what you want to know is what's the median. Well, there's a lot of things you may want to know, but one of the primary ways to analyze this is the median survival. Median means half is less and half is more. So what was the survival where half the people live longer and half the people live less? So you can simply look where the 0.5 is. That's a probability of survival, 50%. Move it over, and you can say you're looking at essentially. I defined it right here: 7.7 versus 6.4 months. The overall survival in 2000 for people with metastatic melanoma at the most, based on this study, was 7.7 months. Now, obviously, you could see there's a tail of the curve. About 20% of people live longer, but if you look at averages, and somebody says, how long can I expect to live? You have to say, look, your life expectancy is 6 to 7 months. And it was a victory because temozolomide's oral, it's less toxic, but it didn't improve overall survival. The response rate, you can see down here, the complete and partial response rate was only 13.5%, no different than the dacarbazine group. So it was pretty grim for people who were newly diagnosed with metastatic melanoma in 2000. Fast forward 10 years, New England Journal of Medicine publishes a manuscript showing that all of a sudden, immunotherapy has a role to play in melanoma. Now, this is a molecule called ipilimumab. Ipilimumab is the first approved checkpoint inhibitor. It targets something called CTLA4. You don't have to remember this, we'll come back to it, but it targets a specific molecule that breaks the immune system. It's the foot on the brake of the immune system. So this antibody interferes with that, and it allows the immune system to function and to seek out the cancer cell. So this study was a second-line study, and because it was a second-line study, there was no standard of care to compare it to, but it was randomized. So what do the authors do? They used a molecule called GP100. GP100 was a cancer vaccine that was being looked at the time, and there was some evidence it might have a role in melanoma. So for something called clinical equipoise, meaning you're trying to give patients a benefit of the doubt of having a control arm when there really was no control arm. And so this was a large study, 676 patients. They were randomized 3 to 1 to 1. So there are actually 3 arms. 2 arms were the ipilimumab arm. One of the ipilimumab arms was combined with GP100. They wanted to see if the combination was better than single agent ipilimumab. And then there was a control arm of GP100. Primary endpoint was overall survival. And this study achieved its primary endpoint. Ipilimumab improved median overall survival compared to GP100. You could see it here in the graph. Look again at our 50% mark. The graphs separate. And if you look at the the actual numbers, how much do they separate? Well, the use of ipilimumab in the second line setting was 10.1 months median survival versus 6.4 months. I just told you 10 years ago that frontline therapy, people only live 7.7 months. All of a sudden in second line, meaning they've already got the benefit of frontline chemotherapy, they're living another 10.1 months. And the p-value was 0.003. Anything less than 0.05 is statistically significant. It's just a way to determine if something could have happened by chance or was a true treatment effect. And we can talk more about statistics if you want. That's not the point of the talk, but I'll stipulate that any p-value less than 0.05 is meaningful. Now, this study also looked at overall response rate as a secondary endpoint. I just showed you that in the frontline setting using chemotherapy, the response rate was 12 to 13%. Usually, as you go further down the lines of therapy, the response rates become much less, treatment effect diminishes. So all of a sudden, in the second line setting, you get an overall response rate of 11% from ipilimumab, which is the same response rate you got in the front line. So this is starting to become pretty interesting. And this central graph or figure is called a forest plot. And it's a way to quickly look at all the predetermined subgroups: gender, age, stage, LDH. LDH is an important prognostic marker in melanoma. Prior use of immunotherapy, interleukin-2, I already briefly talked about that. And if you look at the unity line, number one, anything to the left indicates that all the subgroup benefited from the treatment. Anything to the right suggests that the control arm was better. And you can see in this forward plot, every subgroup benefited from ipilimumab. Now, from the vision of a medical oncologist, improvement from 6.6 months to 10.1 months as median overall survival may not seem so significant or profound for the patient coming in, but it is important. And if you just look at raw numbers, molecules that improve median survival by 3 months are worth $2 or $3 billion to a drug company. So these are very meaningful improvements, and the reason is it's a median. Some don't do well, but quite a few are at the tail end of the curve and doing a lot better. Just trying to understand who those people are. So what I did here is I took the Kaplan-Meier survival curve from the first study on the right, and I juxtaposed it to the Kaplan-Meier survival curve from the ipilimumab study on the left. And I took the liberty to show exactly the median survival at 50% with the red line. So if you look on the right-hand side, you track 50%, the red line to the curve, and you drop it down, you can see the median survival is like 5 months, okay? And that's frontline therapy. All of a sudden, we look at ipilimumab, second-line therapy. We do our same little design, our same little intervention, our same little line, and you could drop it down, and all of a sudden you see you're at 10 months. Now the curves don't look that much different because if you look at the bottom, which is the x-axis, this goes out to almost 5 years. The x-axis on the other study is only 24 months, 2 years. So it's not as magnified, but I promise you could see it. The numbers are very different. The other thing that's really interesting is once you get beyond 2 years, and all these patients on the immune therapy study stopped therapy at 2 years. That's how the studies were designed. Nobody realized the survival, the curve plateaus at 2 years. All of a sudden, there's a group of people, 20 to 30%, after 2 years, don't relapse and are living. Are these people cured? So that was in 2010. Now in 2018, 2 men win the Nobel Prize for Medicine because of their discoveries of checkpoint inhibitors that led to the development of immune checkpoint inhibitor antibodies. And obviously over the past, that preceding 10 years, many new studies, thousands of studies were conducted, literally, looking at a variety of checkpoint inhibitors. There's, I believe, 5 approved checkpoint inhibitors today. The man on the left is from Japan, Tasuku Hanjo. He identified PD-1, which is actually probably much more potent and useful than the CTLA-4 ipilimumab. We'll get to that in a few more slides. The man on the right is Dr. Jim Allison, and he discovered CTLA-4, the other checkpoint inhibitor that led to Ipilimumab, the study we just spoke about. So Dr. Jim Allison has a connection to San Diego. He did his postgraduate work at the Scripps Research Institute, and now he's at MD Anderson, which I've been collaborating with TSRI, and now with Scripps MD Anderson, I collaborate with MD Anderson. So these two men, and there's many others that led to these discoveries, were awarded the Nobel Prize for the profound scientific and clinical impact of checkpoint inhibitor therapy. Checkpoint inhibitors changed cancer medicine. Right now, there's over 3,000 ongoing immune therapy studies that you can see. It actually turns out that more than 2/3 of all cancer studies are incorporating immunotherapy, and it's probably even more than 2/3 now. So, the title of this talk, based on what I just told you, really could be Immunology is the New Oncology. And I'm lucky that my major at UCLA was microbiology, molecular genetics, because it had a lot of immunology. I was taught by one of the giants of immunology named Dr. Eli Surkars, who came down to San Diego to create an immunology institute here in La Jolla. And really, every oncologist needs to become an immunologist today. So we just talked about cutting edge. Really, you guys know a lot about medical oncology, the role of chemotherapy and immunotherapy now, at least in melanoma, and the cutting edge of how immune therapy has changed medicine and the types of molecules that are being used and targeted. The whole idea of immunotherapy started, though, more than 140 years ago, when two men, Bush and Felison, identified that patients that had erysipelas is a cutaneous skin infection, often in kids. They found patients with sarcomas that had erysipelas sometimes had tumor remissions. And it was, they identified the culprit bacteria as something called streptococcal pyogenes, strep pyogenes. Anyways, there's a man named Coley. He is apparently the father of cancer immunotherapy. I think it's a very dubious title for him when you hear what I tell you he did. He took the next step and isolated and grew up streptococcal bacteria and directly infused the bacteria into cancer patients. And out of the 3 patients he injected, 2 died of sepsis. Big surprise. So based on that failure, he modified his approach to heat kill the bacterial organisms and combine and add in another bacteria called Serratia marcescens. And that combination became known as Coley's toxin. Now, if you look to the left-hand side, I found a article in the New York Times, July 29, 1908. And look at what it says. Erysipelas germs as cure for cancer. Dr. Coley's remedy of mixed toxins makes one disease cast out the other. Many cases cured here. Physician has used the cure for 15 years and treated 430 cases. Probably 150 sure cures, probably not. The problem is that none of this was obviously IRB approved. Those didn't come around, IRB boards didn't come around until after the Holocaust. All this sort of stuff was anecdotal and non-reproducible. And so I don't recommend using Coley's toxin at any alternative medicine center that patients may go to. We come back to Dr. Paul Ehrlich. He won the Nobel Prize in 1908. He came up with a concept of immunosurveillance, the idea that cancer cells are always being produced in the body, and we have an immune system that can eliminate them, identify them, and eliminate them. In the 1950s, this idea was expanded by Thomas and McFarlane. Independently came up with the idea that that cancerous cells produce unique epitopes or antigens, little snippets of protein, sugar that they produce that are expressed on their cell surface that allow our immune system to differentiate self and non-self, and then allow the immune system to kill that cancer cell. Okay, so we've talked a lot about the immune system, but really haven't discussed the mechanics. And I think just a few slides to look at a 30,000-foot level down and understand some of the key players. So the left-hand image is of our body. And we think about organs, the heart, the lung, the bone, and these are organs, but the immune system is also an organ, but it's an organ that's diffuse. It's spread out throughout the entire body through lymph nodes, underarm lymph nodes, bone marrow. Our intestines have specialized lymphatic collections, our appendix, thymus, spleen, liver. And there's immune cells in all these independent organ systems. And they serve to survey the body. They serve to help us fight infections when we get bacterial or viral infections. Sometimes they go haywire and produce autoimmunity like lupus, rheumatoid arthritis. The image on the right is drills a little bit deeper into the actual cells. Now the bottom cell is called the hematopoietic stem cell. That's as when we were all fetuses in our mother's womb, hematopoietic stem cells were formed in the liver and the spleen. As we grow, they become limited to the long bones, to the bone marrow. And the bone marrow produces these self-reproducing stem cells. The stem cells then, especially when we were younger kids, would circulate up to the thymus. And the thymus is where they got their education. It was their schooling. Literally, the thymus ensured that the T cells that were produced were able to identify self, which was important. Otherwise, you couldn't differentiate self and non-self. But T cells that were too attracted to the self were eliminated. So it protected us from autoimmunity. Now, there's two basic parts of the immune system, the innate and the adaptive. The innate immune system is part of the parts of the immune system that are ready to go. They got their guns cocked. When bacteria come in, they're ready to gobble them up and kill them. And so those cells that are involved are neutrophils, macrophages, dendritic cells, eosinophils, something called natural killer cells. We'll get back to that in a minute. When I give somebody chemotherapy, chemotherapy kills the white cells, often the neutrophils. So we dose chemotherapy according to what the neutrophil level is at the time. Now, the adaptive immune system it's like the cavalry, so it's the reinforcements. The innate immune system is sitting there, they're holding off the infection, and all of a sudden, and over hours to a few days, the adaptive immune system is learning who the intruder is, and the adaptive immune system is learning how to target those specific invaders. And those include T cells. T cells could be T helper cells or cytotoxic T cells. Those are also the immune system cells that kill cancer. And then we'll come back to the natural killer cells. So natural killer cells, you can see, sits right at the intersection between innate and adaptive. They sit in our liver. The liver is exposed to a lot of our food, to our intestine. And so it's often the first organ that interacts with the outside world. And so it serves to promote both the adaptive and the innate immune system. So all these things are happening in a very complex interplay. Okay. So now, what are checkpoint inhibitors? Well, on the left-hand side is an image of an antibody. An antibody has two long chains and two heavy chains and two light chains. At the tips of the heavy and light chain, they're highlighted. They're less dark. Are what are called variable and hypervariable regions. Antibodies mutate over time in the B cell to become very specific to what is invading our body. Now, these antibodies can be grown in test tubes and be used therapeutically. The first antibody ever approved was something called rituximab, which is actually invented in San Diego with IDEC Pharmaceuticals. And there's many, many of these that exist. So the laboratory is able to grow only the antibody that targets has a specific target. Now, if we move over to the right-hand side, we're looking at three cells. The APC cell, which is called an antigen-presenting cells. These are the Pac-Mans. We talked about them as part of the innate immune system, the macrophage, the neutrophil, dendritic cell that gobbles up whatever it finds in the bloodstream. It gobbles up cells that are dying, and it processes those elements of what it takes in, and then it presents it on the surface. And that's this little blue circle. It's these little snippets that differentiate those invaders or those cancer cells. And that is how our T cell, the CD8 T cell, knows what it needs to attack. Now, you have the tumor cell here that also is displaying what it is producing. Again, often it's separate or unique to the healthy cells. And again, it's the way the T cell can target the cancer cell. But just because a T cell can identify something unique in an infected cell or a cancer cell doesn't mean it's going to act. There's all these other communication signals that can activate it. CD28, that's a co-stimulator signal, or turn it off. Now PD1 or PD-L1, you can see my laser pointer, program cell death 1. That is how our body protects. It's kind of the last gap measure, so that if there's any bad actor T cells trying to attack our body, our healthy cells can still shut them off. The tumor cell is smart. It knows this. It hides from the immune system because it uses that same mechanism to shut off the T cell. So tumor cells express PD-L1. Just because a cancer cell knows it should kill the tumor cell doesn't mean it will. If the tumor cell shuts it off, that T cell is called anergic. It doesn't work. There's another checkpoint called CTLA4. I mentioned that already. That was what the first antibody we talked about, Ipilimumab, targets. This image is of what's called PD-1 or PD-L1, the two sides of the same coin. And there are many monoclonal antibodies, immune checkpoint inhibitors, that block it. And so you could see the image of the antibody. Look at the left-hand side, the big image, and the right-hand side is the same image. That antibody goes in there and it blocks the binding, the interaction of the signaling that turns off. It blocks the turning off of the immune cell. So that's what I mean by it. It allows the immune system, it releases the brake. Without a brake, without the brake being depressed, it can kill the cancer cell. Now, it raises other risks of autoimmunity. These drugs are surprisingly innocuous, but there's certainly risks you need to be aware of. So, all right. So this is from 1994. This was the first immune therapy ever approved, and was approved in melanoma as well as renal cell carcinoma. This was an immunotherapy called interleukin-2. We already mentioned that as one of the cytokines, one of the modalities that immunotherapy for treating patients. And so this was a study of 283 patients consecutive. So it wasn't randomized. Whoever had metastatic melanoma or renal cell cancer were able to be screened and accrued to this trial. And they were treated with high dose bolus interleukin-2. Now, if you look at the outcomes, table 3, I have my laser pointer over it. You can see that 134 patients were melanoma, 149 were renal cell. Among the melanoma patients, the overall response rate, complete plus partial response rate, was only 17%. 17%. And you could see that on the right-hand side, figure 2, the survival, I mean, the median survival, go much or medium, was about a year on this therapy. So how the heck did this get approved? Well, if you look at the people that had complete responses, now in melanoma, 9 patients had complete responses, and renal cell was 10. And you go down to the Kaplan-Meier survival curve, figure 1, the figure right below it, you can see that among the people that had complete responses, almost nobody relapsed, both for melanoma on the left and renal cell carcinoma on the right. If we just look at melanoma, 80% of those patients who had complete responses did not, by 24 months, did not relapse. They were cured. So if we look at the 9 people of melanoma that had complete responses, 80% of them are cured. So 80% of 9 is basically 7. So 7 out of 134 patients were cured. Now, that seems like a really small number, and it is, but for those 7 patients, that's 100% for them, and they're cured. Now, that led to the approval. That also speaks to the fact that there was just nothing available for these people. So you can say, okay, well, why not just give it to everybody? And the issue is interleukin-2 is the cytokine that our body produces when we have infections. It promotes proliferation of T cells, T helper cells, cytotoxic T cells, the ones we just mentioned. And when you get a proliferation of T cells out of control, these high doses, supratherapeutic doses, you get very sick, you get septic. People died on this therapy, and patients were only able to receive this therapy if they had a certain level of performance, strength, and they often had to be put in the ICU for several days to be monitored. They would get incredible amount of fluid retention. And so it was like going through hell to find a small number of people that were cured. So it was an option, but it really wasn't widely used or applicable to the vast majority of patients. Okay, don't worry, we're not talking about politics, but, my friends that know me, my family that knows me may say, well, I put a picture of Jimmy Carter, and you can see where he is. He's in front of what's called Washington Old Hall in May 1977. Washington Old Hall is in a town of Wessington in England, because I like history. And you know what? I've always learned when you give a heavy duty medical talks, especially to people that don't know, read about this all the time, it's good to break it up. And the reality is that they may be partially right, because my family did visit Washington Old Hall when we were in England in 2019. It was pretty incredible. It's George Washington's ancestral home. Happy to talk to you more about it. Always nice to show the greatness of our country sometimes. So I'm happy to talk to you more about it. But there's another reason, and this is the reason. So Jimmy Carter has metastatic melanoma, and he may be cured of it. And in 2015, he had brain metastasis of metastatic melanoma. And so that was 2015. I haven't heard that the man's died yet. And in fact, I've heard that the tumor has melted away. And this is his own statement. After immunotherapy, they didn't find any cancer at all. So this is a perfect segue into going back to our story of immunotherapy and checkpoint inhibitors, because Jimmy Carter received this molecule called Pembrolizumab. And Pembrolizumab is like ipilimumab, is that it is an immune checkpoint inhibitor, but it has a different target called PD-1. I told you guys that ipilimumab's checkpoint target is CTLA-4. So all of a sudden along comes Pembrolizumab, a different immune checkpoint inhibitor. And, there were early studies and they were very promising. And so always in medical oncology, you're saying, okay, well, we got something new. Let's see if it's better than the current standard of care. And the current standard of care, based on what I told you from the study in 2010, where ipilimumab was compared to GP100, was ipilimumab. So this was a study among 834 patients, the biggest study we've spoken about so far, who either were untreated or had only one line of prior therapy. So, many patients were not exposed to a lot of prior therapy. And patients were randomized, this was a phase 3 study, to receiving either pembrolizumab dose every 2 weeks, pembrolizumab dose every 3 weeks, or ipilimumab. That's why it's a 1-to-1-to-1 randomization. There were 2 primary endpoints, progression-free survival and overall survival. And what ended up happening is that the outcome, the results of the people that were dosed at 2 weeks or every 3 weeks was equivalent. So why give somebody a dose every 2 weeks when you get the same outcome dosing it every 3 weeks? So we're going to focus on the Q3 week data. And you could see that the most important outcome always is overall survival, and that's the Kaplan-Meier curve on the right-hand side. My laser pointers over it. You can see that there's an early separation between ipilimumab, the graphs of ipilimumab, and pembrolizumab. And both arms did so well that the Data Safety Monitoring Board, which are required to intermittently evaluate safety and data from every study, stopped the study prematurely because they determined there will not be a median overall survival. Why will not be a median overall survival? Median overall survival is the 50% survival rate. Both these curves are not going to cross that point. In fact, they're plateauing. So they stopped the study and used 1 year overall survival as the justification to approve Pembrolizumab as a frontline therapy for melanoma. And the 1 year survival with Pembrolizumab was 70%. It was 58% with Ipilimumab, which is great compared, would have been great compared to the first chemo, Temodar, temozolomide. All of a sudden, we're getting these incredible survival rates. The p-value, remember, anything less than 0.05 is meaningful. If you look at the progression-free survival, it also met its primary endpoint, but you could see that a lot of people relapse. However, in the Pembrolizumab arm, there may be a plateauing of the curve around 1 year, out around 30 to 40%. Wow. All of a sudden, these are not just baby steps, they're big steps. And if you remember, the response rate with Ipilimumab, the second line setting, was 12%. Pembrolizumab is 33% response rate. That means that 33% had at least tumor shrinkage of 30% or more. Also, Pembrolizumab is a better tolerated regimen than Ipilimumab. So all of a sudden, we have a drug that's better tolerated, more effective. And so it kicked Ipilimumab out of the treatment paradigm. I wanted just to go back and look at the Kaplan-Meier curve from the initial temozolomide study, the first one we ever looked at, that famous red line, 50% survival gives you about a 5-month median survival for Temodar. Now look to your left. All of a sudden, 50% overall survival, neither treatment arm crosses that threshold. It's pretty incredible. So at almost a year and a half, the people that were Pembrolizumab treated, 60% are still alive. At a year and a half with Temodar, less than 20% are alive. All right, like any good medical oncologist, the experts that treated melanoma and conducted these studies said, well, if one is good, maybe two is better. So they combined both checkpoint inhibitor therapies. There was a study, probably other ones, but this is an important one, kind of a pivotal one to refer to, called the Keynote 029 study, where patients were treated with both Ipilimumab and Pembrolizumab. And this was this is called a phase 1B. So a phase 1A is a dose escalation looking for safety. Once you know safety, oftentimes you'll conduct what's called a dose expansion, where you keep that dose, but accrue more patients to get a better sense of safety and a clue to efficacy before you do a randomized study, which costs a lot of money and a lot of patient lives. So in this study, there were 153 melanoma patients, all untreated. The primary endpoint was safety. I told you it's a phase 1 study, that's appropriate. But if you actually look at the efficacy, the overall response rate with the combination was 62%. Let me go back a couple slides. The overall response rate with single agent Pembrolizumab was only 33%. All of a sudden you combine them, the response rate is 62%. And even more incredible, looking out beyond 3 years, 75% of people are alive at 3 years. Now, some of them may have relapsed and had other therapies. So let's look at the PFS, progression-free survival. At 3 years, 60% of people, it says 59% of people are alive at 3 years. Not just alive, but without progression, meaning they were treated and the tumor never progressed. And look at this. And the other point I forgot to mention is these people were taken off therapy at 2 years. Always immunotherapy studies, we could talk about why, are stopped at 2 years. So these people come off study at 2 years. After 2 years, they're just being monitored and nobody's relapsed. So there are probably many people now getting cured of metastatic melanoma with combination immunotherapy. Now, we're going to touch on the role of checkpoint inhibitors beyond melanoma to other solid tumors. This was a key paper published in Science, kind of one of the top 2 scientific clinical journals in the world, looking at Pembrolizumab, which we just mentioned, in a variety of solid tumors. I think they included 12: cholangiocarcinoma, colorectal, endometrial, gastroesophageal, neuroendocrine, osteosarcoma, pancreas, prostate, a whole bunch. You can list, you can see them here. Among solid tumors that had what's called mismatch repair deficiency. Now, we didn't talk about that yet with melanoma because it's not important in melanoma. Melanoma is just a sensitive, an immunotherapy sensitive tumor. Most tumors are not. So it was discovered that tumors that have a high rate of what's called mismatch repair or microsatellite instability are much more likely to respond to immune therapy. And we could talk about what that means. The bottom line is these tumors have a lot of genetic changes. And when you have a lot of genetic changes, they're able to demonstrate or display them on their surface. We can, if you remember that slide I showed you where the tumor cell is interacting with the T cell, they're displaying it. So it gives the immune system many more targets. The problem is they also often express PD-L1 and turn off the T cell. So this investigator looked at a variety of solid tumors that all had mismatch repair deficiency. All of these people had at least one line of therapy. That means many had 2, 3, or 4 lines of therapy, heavily pretreated. The overall response rate was 53%. That's an incredible response rate beyond first or second line therapy. 18 of the 86 patients had a complete response. And in fact, again, let's look at the 50% threshold. This study could not determine a median OS or PFS because none of these lines crossed 50%. If you go to progression-free survival at 2 years, nobody's relapsing. And these people are coming off therapy. So there are subgroups of people with solid tumors that can be, I mean, it's hard to say the C word, not cancer cure. It's hard to say that in metastatic disease, but it's becoming, that word is not as taboo to be used anymore when people have these profound, durable remissions to immunotherapy. Okay. So we already talked about this a little bit. How do you predict who's going to respond to immunotherapy? Well, some tumors don't, it doesn't matter. Melanoma is one of them. Anal squamous cell. There's other tumor types that some lymphoproliferative subtypes don't matter. They're just more they're more sensitive to immunotherapy for whatever reason. In other tumor types, there are a variety of predictive markers that need to be looked at to make sure it makes sense to treat patients. Now, there was a paper just published in Cell a few months ago that suggests that something called tumor mutational burden is the most important predictive marker. Again, harking to the idea that the more mutational changes a tumor has, the more likely that it'll respond. So that's one. Microcell instability, PD-L1, talk about PD-L1 as a brake on the immune system, something called EBER that's more specific for esophageal adenocarcinoma. So if somebody has metastatic cancer, you have to get broad genomic profiling that includes these markers. And patients that can qualify, that have these right markers, immunotherapy needs to be, if not the primary focus, the second line therapy, but usually the primary therapy because it's so significant. So much of medical oncology now is trying to understand how do you convert those tumors that are immune cold and turn them into something that's immune hot? How do you give a tumor that doesn't have these markers immune therapy and make it effective? So I took a screenshot of the package label for Keytruda, also called Pembrolizumab, and you could see the incredible approvals, indications that it's approved for: melanoma, non-small cell lung cancer, small cell lung cancer, head and neck squamous cell cancer, classical Hodgkin's lymphoma, primary mediastinal large B-cell lymphoma, urothelial carcinoma, which is bladder cancer, any microsatellite high tumor. We just showed you why that is. Gastric cancer, esophageal cancer, cervical cancer, liver cancer. That's another one that doesn't require microsatellite status. Merkel cell carcinoma, renal cell carcinoma, endometrial carcinoma, any tumor mutational burden high tumor. I just showed you why. Cutaneous squamous cell carcinoma, triple negative breast cancer. And that only covers the approved indications. Medical oncology moves way faster than the government and the FDA. Some pharmaceutical companies never you want to invest the cost to get a formal approval, but if a clinical study shows benefit, you can use it off-label. But there are more than a dozen indications today that are approved for immunotherapy. Now, I'd love to end this talk on the highest of a high note that I think we're at now, but there's a lot of work that needs to be done, mainly in tumors that I treat, specifically pancreatic adenocarcinoma. Checkpoint inhibitors have almost zero benefit in treating pancreatic cancer patients. This really was one of the studies that put the nail in the coffin of checkpoint inhibitor therapies for pancreatic cancer, at least until there's a different strategy, newer combination therapies to be looked at, and they are, and I'm happy to talk to you about them, and I'm excited about them, and we're involved in some of those studies. But this was a study looking at patients that had progressed after one line of pancreatic cancer therapy, usually Gemzar and Abraxane or something called FOLFIRINOX. And you could see that patients were randomized to either one type of checkpoint inhibitor or two types. Now, we haven't mentioned these before. So there's something called Dervalumab and Tremelimumab. They're different, made by different companies. They're the same idea. Tremelimumab targets CTLA-4, like Ipilimumab, and Dervalumab targets PD-L1, very similar to Pembrolizumab. So, I mean, at the end of my day at work, my tongue is tired from these gymnastics of words, but you could trust me that these are checkpoint inhibitors. And you can see that the progression, first of all, the two curves overlap completely. There's no difference in outcome whether you do one therapy or the combination. And there's the progression-free survival is terrible. Within 1 or 2 months, people are progressing. The median progression-free survival is probably 1.5 months, 1 month. The median overall survival with second-line pancreatic adenocarcinoma with checkpoint inhibitor therapy is 3 months. That means half the people are not alive anymore by 3 months who receive this therapy. This is what's called a spider plot. The spider plot is a way to look at responses per patient over time. So each line is an individual patient, and if the line goes up, that means the tumor's progressing. If the line goes down, it means it's responding. A true response for a clinical study means a tumor has to have shrunk by 30%. There's only one responder out of all these patients. Only 1 out of 65 patients responded. That's why you get a response rate of 3.1%. And that's bad even for pancreatic cancer. So there was another study that was presented as an abstract. It didn't look good on a slide, that combined Gemzar plus Abraxane, potentially effective chemotherapy with immune therapy. And again, there was no benefit. So in today's world, the standard of care for pancreatic cancer is no immunotherapy, unless you're among the 1 or 2% that's lucky enough to have microsatellite instability. Again, only 1 to 2% of pancreatic cancers could benefit. But there are a variety of clinical agents that are being looked at in phase 1, phase 2 studies that look very interesting. So why aren't they benefiting? Pancreatic cancer is the poster child for stroma. We haven't really talked about that, but I just wanted to briefly show it to you. Stroma is not the cancer cell, it's all the stuff around it. Now, this is what a normal pancreas looks like. It's healthy, there's a lot of cells, there's architecture. Pancreatic cancer is really a chocolate chip cookie. If you think about the cancer cell as a chocolate chip, okay, these are the cells or the nuclei are, but the vast majority is this pale collagen connective tissue, hyaluronan, immune cells, and the vast majority of the tumor is not even cancer. It is this reaction that the body forms around the cancer. And within that stroma are something called myeloid-derived suppressor cells, regulatory T cells. These cells populate many cancers and shut off cytotoxic T cells. They shut them off, and it doesn't matter if you give them immune checkpoint inhibitors. The impact of these immune suppressor cells is too much. Now, a lot of novel agents are being looked at to modulate these inhibitory effects of these other myeloid and regulatory T cells so that they don't turn off the good fighters, the troops. But, time will tell, and the studies are ongoing. Okay, so I just wanted to show you a few examples of really incredible stories. These are all de-identified CT scan images. This is of an 89-year-old woman I take care of with what's called neuroendocrine tumors. Neuroendocrine tumors, I've actually had a few publications on that. They're kind of odd, somewhat mysterious tumors. This is an image on the right-hand side from March 2020. This is the woman's liver, and all these black circles, masses are cancer. I mean, her tumor, her liver was full of cancer. And she had progressed on standard of care chemotherapy. She was 89. How much can I do to her? Didn't want to kill her. And I said to her, look, there was just a brand new study that was published called the DART trial, and it used combination checkpoint inhibitor therapy, just like we've been talking about. And it said, look, the response rate was 44% in people with high-grade neuroendocrine tumor, just like you. And that was in beyond frontline therapy. I said, look, I'm willing to give it to you. Are you willing to get it? So she went ahead and got it, and she is in an essential complete response right now. This is the image that we took in February 2021. It's almost a year from the image showing all the tumor. We started her on combination checkpoint inhibitor therapy. After a few cycles, based on the way the protocols were written, we removed one of the agents, and now she's only on one drug, Pembrolizumab. And when she gets closer to 2 years, we'll talk about stopping all the drugs. So this is a woman who would not be alive right now, and she's having 0 side effects. This is a really interesting example from 5 or 6 years ago of a woman who had metastatic esophageal adenocarcinoma. She was on a clinical study. This is a clinical study I had with a PD-L1 targeting agent. And this is an image of her liver. And this is a tumor deposit in her liver. She started on the study in June 2015. The study required a CT scan imaging every 6 weeks. We do a CT scan 6 weeks later, and all of a sudden, this tumor, as well as others throughout her liver, but I thought this was an easier image to look at, was growing. And I said, look, you know, I'm so sorry, these are the scans, the tumors are growing, but I said, you know what, but I want to keep you on the study. And she goes, what are you talking about? I said, how do you feel? She says, I'm feeling better. My appetite's coming back, my weight's coming back. Her tumor markers were coming down, the blood markers we use to kind of get a peek, a clue to what's happening. But she goes, but my tumor's getting bigger. I said, you know what? I think this is something called pseudo-progression, and it's a well-known phenomenon in immunotherapy. It's not common, but it happens. You have to be aware of it. Sometimes when you get effective immune therapy, the tumors get bigger before they get smaller. And it's not that the tumor's getting bigger, it's that the immune system is invading the cancer deposit and producing inflammation and the tumor swelling. She goes, okay, Dr. Siegel, you seem like a nice guy. I've only known you 6 weeks, but I'll trust you. So we kept her on the study, we kept going, and we scanned her 6 weeks later. And all of a sudden, that tumor almost completely disappeared. And so you just need to really understand the way tumors respond to all these new therapies, and sometimes bite the bullet and keep going, especially when somebody's clinical status is improving. So the last image I have is a man in his 50s, really nice guy, also metastatic esophageal adenocarcinoma. I started him on chemotherapy, FOLFOX chemotherapy. He was having some progression. At that time, you know, this is a few years ago now, we got all the biomarker testing, the microsatellite status. He was microsatellite high. I said, listen, let me put you on Pembrolizumab. It's a well-tolerated therapy. You know, I think there's a good chance you could respond. Let's see what happens. So this is a CT scan on the right from August 2018, 2.5 years ago. And this is an image of the chest. And this hot dog looking structure is called the aortic arch. It's your aorta as it loops back down, where the ascending aorta loops down to become the descending aorta. This is your trachea. This is his esophagus. These black spaces are the airways, normal looking, the lungs. This is the superior vena cava, and this is the beginning of the carotid arteries. So these are all normal structures. These two nodes, these two spots are not normal. These are metastatic cancer deposits in tracheal lymph nodes, and they're pretty large, like probably 1.5 to 2 centimeters. And if they got bigger, it could start impinging on the airway. So we put them on immunotherapy. This is August 2018. Fast forward more than 2 years, 2 years and 4 months. And look at a CT scan now. Same cut, I got the same cut, and these are gone. There is zero evidence of cancer in his body. He had a complete response to Pembrolizumab. And look at this, it's really interesting. He started Pembrolizumab in August 2018, right when the CT scan on the right-hand side was taken as the baseline. We stopped immunotherapy. Like I told you happens in all these studies. We stopped it less than 2 years later. We thought he was having some side effects from the immunotherapy. They ultimately were not related, but we still stopped it. We stopped it in February 2020. Okay, February 2020. This is a CT scan from December 2020. Almost a year later, he's been off all therapy. He's in a complete response, no relapse. And in fact, just saw him recently another scan. I scan him every 3 months for metastatic cancer, no relapse. Is this guy cured? Maybe. Okay, I've done all the talking, as I always tell my patients at the beginning. Now it's your turn. So, before I open up the floor, I just wanted to reserve, I promised my close friend, Marcy Leiserbram, she could ask the first question if she wanted. I'm sure my mom has a question too. Otherwise, everybody is a free-for-all. I'm happy.
So actually, yeah, so what I'm gonna do is I'm gonna say that, so Marcy for sure can go, and obviously, Mom, if you have a question. What I'm gonna recommend people do is if you have a question, please raise your virtual hand, and then I will go through and call on you in the order that the questions come in. In order to raise your virtual hand, for most of you can find it under the reactions tab on the bottom. There's a little thing that says raise hand. If you have an older Zoom, then you might find it under participants in there, but it will say raise hand, and that's what you want to do, not the clapping one, which we'll go in a minute. So Marcy, you get to go first. You can unmute yourself and go ahead.
I don't have any questions.
Okay.
Sorry.
No questions.
Hold on, I'm gonna
right.
I'm gonna stop. Marcy, did you have a question that you wanted? Otherwise, I'm gonna go
just
through.
joking around with Marcy.
Oh, okay, fine. All right, well, let's go. Wayne has his hand up. Go ahead, Wayne.
Sure. Thanks, Darren. Really appreciate that. Really great talk. I was curious to know, with respect to pancreatic cancer, we always hear that early intervention is best. And are there ways to detect pancreatic cancer early? Like, we have colonoscopies for the colon, et cetera. Is there anything going on with pancreatic cancer with respect to early intervention like that and early detection, I suppose?
So I think that's a great question. There's a lot of screening tests for colon cancers, breast cancer, but pancreatic cancer, very similar to ovarian cancer. It's one of these insidious tumors that can grow silently, actually for years. That's a misunderstanding. It can be developing over a long time, which lends itself potentially to screening tests. The problem is there is no screening test. And there's definitely no screening test for low risk or the general baseline population risk. In people that are high risk for pancreatic cancer, 1 or 2 first-degree relatives, or people that have certain genetic syndromes like BRCA, which we think about as breast cancer, but it's also with pancreatic cancer or Lynch syndrome, there are things that can be done, routine MRIs, endoscopic ultrasounds, but it's really unclear how much of an impact they have for early detection. There are some multiplex protein markers in the blood that are not standard of care that are being looked at. In fact, I have had a study where we've identified a circulating myeloid-derived suppressor cell that we think might be interesting, but the point is that there's really no good screening test that's been shown to change outcome. The problem with pancreatic cancer is that even if you detect it when it's very small, it can spread very early. So by the time the pancreatic cancer is detected, it was probably growing in your body for 5 to 10 years, and the metastasis or the micrometastasis already spread a few years before that. So if you have somebody who has localized pancreatic cancer, which only is 20% of people. Most people that present with pancreatic cancer are either metastatic or unresectable. So only 20% can get to surgery, can get to be cured. you get to surgery and you do chemo before and after surgery, 60% of people are still going to relapse, unfortunately. That's a lot better than surgery alone, which is 90% of relapse. But the point is that it spreads early. And to this point, at this point in time, there's nothing that I could tell you is a standard of care that's been shown to change outcomes.
Thank
Wow.
you. Thank you.
Thank you. Are there other questions? Please go ahead and raise your hand if there are other questions.
I have a question.
Okay, you didn't raise your hand, Claire, but I'll let you come in.
We couldn't find it.
It's not appearing on my bottom
Okay,
right.
go ahead.
No, that was so interesting. Thanks so much, Darren. I was just curious, it kind of piggybacks on the previous question. Do you envision immunotherapy being more effective if you can treat earlier in the disease process?
That's a great question. I mean, there is a concept of the idea that, at least for chemotherapy, when you administer chemotherapy, you're going to kill a certain percentage of cells. You're going to kill this 50% of cells. So if you treat earlier, you're going to, that the absolute number of cells may be less, but the percent's going to be the same. So you have a better chance of maybe killing micrometastatic disease that may have spread that we can't see, that's going to ultimately be the source of relapse. In immunotherapy, all the early studies of immunotherapy were used beyond the front line. So people had already been pre-treated. So by definition, these people are already more advanced. Now, that being said, more and more studies are looking at immunotherapy in the frontline setting. And so that's the best approach to get a cure. Now, I just showed you some examples where people that I've treated and people on studies go into deep remissions and don't relapse for more than 1 or 2 years, and maybe they are cured. I think the general point is that, yes, I agree. I think the sooner you can treat metastatic disease with these types of therapies, probably gives you a better chance of being cured. Now, chemotherapy, that's somewhat of a different discussion, but generally when you have less tumor burden, every area that spreads you could look at as a totally separate disease, because every separate clone of cells may have their own specific genomic drivers and acquired mutations that are the source of future resistance. In chemotherapy, it's not clear if you treat early or late, if you're going to change their ultimate survival. So if somebody is asymptomatic with low volume metastatic disease, you may say, hey, you may not progress for 6 months or a year. Why treat you now with side effects? But immunotherapy is different because it's pretty innocuous for most people, and it really changes the biology, and it stimulates your own body's immune system to promote the immune surveillance that was postulated in the 1950s. So I agree. I think everything is going to be moved more to the front line.
So we're now starting to get a few more questions coming in. The Cullys have their hand up, so I'll let you go, and then I've got 2 from the chat that I'll share with you. Cullys are first.
Thank you so much for this very fascinating talk. Getting back to what Wayne was talking about for screening tests, Dr. Siegel, what about doing FISH studies? Is that anything that can be looked into?
For screening?
Yes.
So FISH is a technique, fluorescent in situ hybridization, that it uses hybridization of 2 antibodies on the chromosomes, and you can actually identify when there's genetic alterations, translocations, and it's very specific but not sensitive unless for particular diseases. So what that means is that if you don't have that disease or that specific mutation, it's not a great test, because for a screening test, you want a test that's very sensitive. And what that means is it's going to detect a very small number of false negatives, but you're going to pay the price with some increased number of false positives. And so then once you have a screening test that's very sensitive, then you may want to go and do the FISH test that's more specific to ensure that you're not detecting, telling somebody, oh, you have cancer, and they just have pneumonia or an infection. So, if you looked at specific types of genomic changes that are translocations, like Philadelphia chromosome, c-KIT, in GIST tumors or CML, it might be a useful test to screen, because in those specific tumor types, it's very specific. But there's a lot of barriers but the nature of that test is not ideal for screening. And that being said, if you look at other screening tests like multiplex next generation sequencing, looking at something called KRAS or P53, these are genetic changes that are very prevalent. The most common genetic changes, like in over 50 to 60% of tumors, are going to be KRAS or P53 mutated. Even in that setting, it's not clear how useful it's going to be, because people that are asymptomatic with clear CT scans, you may have a certain population that is producing these mutational changes, and it doesn't necessarily mean they're going to go ahead and get cancer. Obviously, I'd be worried if it was detected in somebody, but because there's not a clear correlation, and because it could be potentially too sensitive, it makes it very difficult to kind of broadly screen the patient population.
Thank you.
So we've got two questions from the chat, and I'll read both of them to you, and you can decide which order you want to answer them. One from Susan, as she mentions the fact that colonoscopies are now being recommended at 45 years instead of 50 years of age. Are we starting to see younger patients with colon cancer? That's question number one. Question number two is really a kind of a COVID and modern related question. Ben asks, can we produce vaccines to prime antibodies for cancer cell markers with the mRNA technology, kind of the way that we're doing in COVID-19? So I'll let you deal with those how you want to.
Sure. So colon cancer screening is at 45 now. And absolutely, there's a whole phenomenon of younger people getting colon cancer. And I've seen it. I've had patients in their 30s, 40s is not uncommon anymore. Traditional age of, the historic time to start screening was 50. But people in their 30s and 40s are getting colon cancer. And it's usually a descending or left-sided colon cancer. Nobody really knows why. It could be from caloric intake in the diet, something environmental, but it is happening, and so I think it's really important to get a colonoscopy at 45, and if you have any risk factors, even at 40 or 35, depending what those risk factors are. In terms of cancer vaccines, people have looked. There's many cancer vaccines are a very hot area. I don't believe there's any approved cancer vaccines today, at least in solid tumors. There's 0 that are approved. You could look at a molecule called, a treatment approach called Provenge, it's called Sipuleucel T. It was approved, I think 10 to 15 years ago for prostate cancer. And patients would have their white blood cells, the patient would donate their white cells. They would be, the dendritic cell would be purified and modified, and then insert back into the patient to promote an immune response. The problem with cancer vaccines is the same problem as with checkpoint inhibitors. You still are going to have those checkpoints and those profound immune suppressive influences in the stroma. Now, cancer vaccines may have a role in conjunction with checkpoint inhibitors and as well as cell therapies like CAR T and NK cells. But for the same reason, cancer vaccines haven't been as effective in solid tumors. CAR T cells, which we may have heard about in the news, very effective for lymphomas, have had very little to no benefit in solid tumors, especially pancreatic cancer, even if you're able to identify the antigen, often something called mesothelin-1, CEA, PMSA, and grow up cells that target that specific antigen. It just doesn't have a huge impact. There needs to be further innovation and strategic optimization with additional agents that can go in and change the biology of the soil of where the cancer is sitting.
Wayne, you have another question.
Thank you. And then just with respect to the likelihood of even developing cancers, of course we know about heredity and you mentioned environmental. And so with respect to environmental, are there things people can do just in general to like in their nutrition or whatever their environment to reduce the likelihood of these types of cancers?
So, I mean, that's people take a lot of vitamins. It's a common question. So I'll give you a story. So people come in, they want to take a ton of antioxidants, CoQ10, vitamin E, fish oil, whatever it is. And I've always had an impulse to avoid it because the way cancer cells die, and I'll get to your question, but I want to just kind of introduce it. The way chemotherapy and radiation works is it produces, they induce oxidation. One of the mechanisms for cell death is by causing free radicals they generate free radicals. Those free radicals go and damage the tumor DNA, and the cell dies through apoptosis. Theoretically, and there was a lot of expert opinion about this, if you take high doses of antioxidants, how do you know you're not going in and rescuing the cell by soaking up all those antioxidants, all those oxidants from the antioxidants that you're taking? So last year, there was a paper in Journal of Clinical Oncology looking at breast cancer patients who were getting chemotherapy. And they looked at patients who were taking supplements before and during chemotherapy and comparing them to people that were just getting chemotherapy without the supplements. And it turns out the people that were taking the supplements actually lived less long. Now, there's not a scientific model that came out of that, but it reinforces the general feeling that high dose of antioxidants are probably not helpful to you when you're on anti-cancer therapy. Now, if we take that to prophylaxis and preventing cancer, you know what? It's just unclear. I can tell you, evidence-based medicine for colorectal cancer, vitamin D is very important. I always tell all my patients that you want to be in the upper quartile of vitamin D, 75 to 100 units. And I have all my patients take take vitamin D. There have been studies, randomized studies in colorectal cancer. It's called the Sunshine Trial, where there was two groups. Both groups got the same chemotherapy. Half the group got high dose vitamin D. The other group got very low dose or no vitamin D. And just that difference in intervention improved median survival by 6 months. So clearly, vitamin D has an impact. And vitamin D is being used. There's something called paracalcitol. It's being looked at to actually treat cancer. So I think vitamin D, obviously, you don't want too high dose, but vitamin D is really important. Vitamin C is probably very important too. Now, Linus Pauling took grams of vitamin C, but vitamin C, especially when you have cancer, may actually function as a chemotherapeutic. And there are randomized studies ongoing in pancreatic cancer and probably other cancers looking at high dose vitamin C, intravenous vitamin C. There was a retrospective study looking at colorectal cancer patients, people that had surgery, had postoperative chemotherapy called adjuvant therapy, and they retrospectively went backwards to see, well, which groups did better, which groups did worse. And what was found is that people that took a certain amount of tree nuts every week actually had a 42% reduction in the risk of recurrent colorectal cancer. So I tell all my patients, look, eating tree nuts is not going to harm you, and there may be a benefit. Now, retrospective study is less strong. The data is weaker, but again, weak data, no toxicity. So, I eat a lot of tree nuts every day because of that. And I think there clearly are fatty molecules in tree nuts that, may be having an impact that we don't understand yet.
So
go ahead.
I'm just going to say, so we're at 11:30. I'm going to just ask two more questions. I know people, we're welcome to stay longer if people want to, but I do want to let people go if they have other things to do. One question from the chat. Gary was asking what your thoughts are on gene therapy, gene editing as a possible cancer therapy.
Well, there's this whole technology called CRISPR, where there's an ability to go in and apparently splice out specifically diseased genes in the living organism and insert healthy genes. I mean, I'm not pretending to be an expert in that. I'm not. But gene therapy, the ultimate gene therapy is actually just giving somebody a new bone marrow when they have an autoimmune disease or leukemia, which is being done. Gene therapy as a therapy for solid tumor cancer is, I'm unaware of any data that involves it. My understanding is it's very hard to enable that gene to be inserted in all the tumor cells that could have spread throughout the body. I mean, there may be some studies looking at it, I just couldn't comment too much about it.
So, and the last question, this actually question comes from me. You know, when I'm speaking with people, and you and I sort of, whether it's actively or inactively, work together sometimes on this, right? And when I'm speaking with people, part of the calculus that they're thinking about when they come to you has to do with how they term in terms of quality of life. And, you know, one of the big fears of cancer treatments for many, many years is just because of the toxicity of the chemotherapy and stuff like that is, okay, I may live an extra 3 months, but do I want to live those extra 3 months?
Right.
So I guess I wanted to ask you, as you're starting to see these immunotherapies and these new treatments, what is the tolerance level of these, and how is that changing the calculus for people, or are there still, is this still issue? Because I think people want to know that.
Yeah, no, it's a great, it's the key discussion I have with every new patient I meet. And I always say, look, there's a risk of doing something and a risk of doing nothing. The risk of doing something is the risk of the chemo. The risk of doing nothing is tumor progression. And even with standard chemotherapy, I see that as palliative in a lot of ways, because there are so many effective anti-nausea medications, energy stimulant. There's so many things that can be done today to overcome the toxicity. And ultimately, if it's effective, if the tumor responds, you're going to be feeling better. Now, as it relates to immunotherapy, these drugs are shockingly innocuous. Now, we talked about two types, CTLA-4 and PD-L1. CTLA-4 is more toxic. There's no doubt about it. The fear is autoimmunity, myocarditis, nephritis, pancreatitis, hepatitis, colitis. Your immune system can attack any part of your body. Now, if you look at PD-L1, the Pembrolizumab, that's the one that's used probably the majority of time. The risk of serious adverse events is probably about 3%. And in that setting, there's ways to turn off the immune system with high dose steroids or other immunomodulatory agents that are used in autoimmunity that are being used in this setting as well. Now, when you do the combination, which I showed you is much more effective in some settings, it's more toxic, but actually the doses have been dialed down so that, and the second drug is stopped after 4 cycles generally. So even then, it's actually much better tolerated. So most people have minimal to no side effects, and most people feel better because they're responding. Most common side effects are going to be fatigue, muscle weakness, sometimes skin rash or thyroid dysfunction that are all generally easy to manage.
Okay. Dr. Siegel, thank you so much. Darren, it's awesome. This was an amazing talk. So much information and actually a lot of hope, which is nice. We understand it's not all the answers aren't there yet, but to see your excitement in what's going on and what the possibilities are in some of the most insidious diseases that we have and that we're dealing with gives a lot of hope. And I'm really happy that you were able to share this with us.
My pleasure.
Ladies and gentlemen, thank you. Thank you for joining us for this CBA Talks.
Thank you.
And we look forward to seeing you in about a month for our next CBA Talk with Jake Hulley.
Bye.
Thank you.
Get off.
Thank you so much, Dr. Siegel.
That was a great talk.